What is up, guys, and welcome to The Prodigal Believer. I am your host, PB, and welcome to another episode that is an interview with another special guest I have on the podcast today. My guest today is going to be talking about intelligent design. He is an evolutionary biologist, and I'm not going to do the introduction. I'll let him do the introduction himself. So why don't you tell us a bit about yourself?
Well, thanks so much for having me on. I'm Jonathan McLatchie, and I currently work in Boston, Massachusetts as an assistant professor of biology at a Christian college called Sattler College, which is based in downtown Boston. And I completed my PhD back in January of this year in evolutionary biology, studying the evolutionary origins of the eukaryotic cell cycle. And prior to doing my PhD, I did a couple of master's degrees, one in medical and molecular bioscience, which I did at Newcastle University in the UK. And I also did a master's degree at Glasgow University in evolutionary biology and systematics. And prior to that, I did my bachelor's degree at Strathclyde University in Glasgow in forensic biology. And I'm currently working on a Master of Arts degree at Southern Evangelical Seminary in Biblical Studies. I'm very interested not just in the science and scientific apologetics, the case for intelligent design, and that type of thing, but I'm also interested in biblical apologetics and New Testament historiography, the case for the resurrection, Christology, the Trinity, that kind of thing as well. I also have a website, which is jonathanmclatchie.com, where I host many of my articles on apologetics and theology and science and so forth. And you can find a link to my YouTube channels there as well. And I also run a service for Christians who wrestle with intellectual doubts concerning the veracity of the Christian faith. So I have a form on my website that Christians can fill out and tell me about their intellectual doubts concerning the Christian faith. And that would include Christians who are really struggling with doubts and whose faith in many cases is holding on by a thread, or atheists or skeptics, seekers who have honest and sincere questions, who want to talk, and also Christians or ex-Christians who recently lost their faith. I want to explore whether their reasons for loss of faith were truly rational, and try to help people to work through a protocol for dealing with doubts in an intellectually responsible way, and also engage with their concerns that they have concerning the veracity of the Christian faith. I also do debates. I've done somewhere between 40 and 50 moderated debates, and I debate atheists and Muslims and people of other faith perspectives as well, including Jews and so forth. So I have quite a well-rounded interest in apologetics and science.
That is quite the record there. I was never a science guy, so it's really great to have an expert such as yourself to come on here and actually truly explain the science from a dignified position. Seeing as you've had your PhD in evolutionary biology, you've had all of these different degrees. It's really great to have you on the podcast, Dr. McLatchie. So, were you always a Christian?
I became a Christian. I was raised in a Christian home. I became a Christian in 1996 when I was age 6 or 7. Just about to turn 7, and on the 3rd of March. And my parent, I was raised in a Christian home, as I said. My father was an elder in the church I grew up in, so I grew up in the church and became a Christian at quite a young age, and then got baptized about a decade later in 2007, I think it was, June 2007, and went to university in 2007 as well. And really, that's where my interest in apologetics and also the scientific case for design and scientific arguments for God took off. I was blown away studying the molecular wonders of the cell and the engineering prowess of macromolecular machines, and often baffled and bewildered at how anyone could go through a university program in the natural sciences and come out an atheist at the other end, because the evidence from the natural sciences seemed to be so compelling for the truth of theism, not just in physical sciences, such as the fine-tuning of the laws and constants of our universe and so forth, in fact, the universe has a beginning, etc. But also in the life sciences, the case from information content, genetic code, and so forth. And during my university years, I also interacted with a lot of people who did not share my worldview perspective. So atheists, Muslims, I knew some Mormons, Jehovah's Witnesses. I had a close friend during my undergraduate years who was a Taoist. And so I interacted with people of different faith perspectives and none. And so became very interested in the question of how we can adjudicate rationally between worldviews. How do we know that Christianity is true and these other worldview perspectives that have adherents and followers, which hold to it with equal arduousness and vehemence as many Christians? How do we know that Christianity is true and those other worldview systems are false? And so that's where my interest in the rational, evidentialist defense of the faith took off and came from.
Nice, awesome. And you said you had a website and you're running a forum for people who are doubting their faith. Do you have any other current or future projects in mind that you wanted to shout out?
Sure. So I have a YouTube. I have two YouTube channels. One of them is my name, Dr. Jonathan McLatchie, and the other, and I host I put many of my debates, interviews, and talks up there. And I have another YouTube channel which is called Apologetics Academy, which is where I run interviews and with various scholars and thinkers from across the theological spectrum. Most of my guests are Christians, but I also have non-Christian scholars, including atheists and Muslims. I've even had a Mormon on. And the reason for that is because the philosophy from which I operate is that if Christianity is true, which I think there's very good reason to think that it is true, then we have nothing to fear from hearing the very best and most erudite, most robust scholarly criticisms of the Christian faith. So I have no qualms about bringing on some of the top critics of the Christian faith to have a discussion and to present their reasons for disbelief. So I run the Apologetics Academy. I used to run it every week. But of late, I've not had so much time, so I run it occasionally. Most recently, I had on John Steingart, who your listeners might know from the Christian rock band Hawk Nelson. He recently came out as an atheist. He no longer believes in God.
Right,
and,
right.
He announced this on Instagram, and he's done a number of interviews. He did a couple of discussions with Dr. Sean McDowell from Biola University, who's based out there in California, and one on Unbelievable, and then another one on Sean McDowell's YouTube channel. And he also did an interview on Seth Andrews' podcast, The Thinking Atheist, and also he's been on a few other shows. At Southern Evangelical Seminary, he had him doing a live discussion with Alyssa Childers, who was also a Christian music artist who went through a period of doubts and is now a Christian apologist. So I had him on my channel to discuss his reasons for doubt and to gently push back on some of the issues that he was concerned about. And in terms of other projects I have, obviously a lot of my speaking engagements are and travels with my apologetics were put on hold by the virus, which is unfortunate. Right. So a lot of my stuff at the moment is happening online, but that's just most of what I'm doing at the moment.
Sounds good. Even with the pandemic going on, it's definitely a good thing that people are able to connect over the internet and have these sorts of conversations. Everybody's stuck at home, so I'm pretty sure everybody has enough free time to start learning new things, as am I doing right now, by talking to you. And for those of you who are not aware, this episode is going to be about intelligent design. Now, intelligent design is essentially a theory of the origins of life, and it has to do with evolution. And the theory of intelligent design has sort of come back onto the main stage of science and of evolutionary biology by people such as Stephen Meyer at the Discovery Institute. You have people such as David Berlinski, the mathematician, the prominent philosopher who studied all sorts of fields, who has also sort of helped float the boat of intelligent design in recent years, and people such as Dr. McLatchie himself. And not a lot of you probably know what the theory of intelligent design is. And myself being not really a science person, can't really comprehend the complexities. So, Dr. McLatchie, in your idea, what is a layman definition, or what is a layman way to help people understand the theory of intelligent design?
Sure. So, intelligent design is basically the study of patterns in nature which bear the hallmarks of an intelligent cause. So, it's the idea that we can infer from patterns in nature that there are some features of living existence that are best explained by an intelligent cause rather than a stochastic process involving chance and necessity. So, Charles Darwin pioneered his theory of evolution by natural selection, and he basically championed the idea that natural selection could explain the appearance of design in the world without recourse to a designing intelligence. Richard Dawkins will say that Charles Darwin made it possible to be an intellectually fulfilled atheist, because prior to Darwin, you looked at living organisms and you saw that they are tremendously complex and give every appearance of being designed for a purpose. And Charles Darwin came along, of course, and challenged that idea by arguing that natural selection could really explain this appearance of design in the world without recourse to a designing intelligence. So intelligent design then would compete with that and argue that actually things appear to be designed because they really are designed. And we would argue that natural selection and random variation, which Charles Darwin, of course, didn't know what mutations were, but we now know that variations in population are caused by mutations. And I would argue as a design theorist that although these processes have some explanatory power, they explain some phenomena about the world. They don't explain the designoid characteristics of life. So the theory of evolution really consists of 6 main components or elements, and you can talk about evolution in the sense of the change in allele frequencies in a population over time, right? An allele is a gene variant, and new alleles are caused by mutations that occur in genes. Mutations are basically copying mistakes, copying errors as the DNA is being copied. So that's uncontroversial. I mean, everyone knows that allele frequencies change in a population over time, and that's in response to environmental pressures, because certain combinations of gene variants help the organism to survive better in its environment than other combinations, etc. Another meaning of evolution is the idea of speciation, which is the idea that you could have a species and you have a population of organisms belonging to that species, and the populations become somehow reproductively isolated or separated from each other, so they can no longer interbreed with each other. Perhaps there's a geographical barrier, for instance, that's set up and established, and so they're no longer able to interbreed with one another, and eventually over time they diverge to such an extent that they, even if you brought the populations back together again, they couldn't interbreed with each other to produce fertile offspring, and they're considered to be separate species. So that's the idea of speciation, which I would argue is also a real phenomenon. Speciation is a thing. And then you could also talk about evolution in terms of common ancestry. The idea that all humans alive on the planet today are descended from a common ancestor, right? And eventually, if you wanted to take a life back far enough, you wouldn't just find that all humans have a common ancestor, but you'd find that all organisms alive on the planet have a common ancestor, right? So you and a chimpanzee and a banana and a frog and a grapefruit and the fungus growing in the back of your foot, these are all descended with modification from common ancestors. And eventually, as you wind the tape of life sufficiently far back, you come to a universal progenitor of all life that we see around us today. So that's the idea of common ancestry. And then there's the idea of gradualism, which is the idea that evolution doesn't happen by sudden leaps and bounds. You don't have you don't have a chimpanzee, for example, giving birth to a human being, right? That would be kind of a saltation, if you will, a very sudden jump. But that's not the way that evolution works. It's a very gradual process that happens over hundreds of thousands, even millions, even hundreds of millions, billions of years, right? So it's in terms of gradualism, you could never really talk about a first human, right? In the same way that it's very difficult to talk about the day upon which you became an adult, right? It's rather arbitrary to say that, okay, so on your 18th birthday you become an adult. Well, that's kind of rather arbitrary, right? So it's a very gradual process that happens that you barely notice, except in the long run. Likewise, how many facial hairs does it take on the human face to qualify as a beard? Well, it's very difficult to quantify exactly how many facial hairs are required. So in an evolutionary scheme of things, there was never a first human as such, but it's a very gradual process as the population changes over time, driven by environmental pressures. Then there's the idea of natural selection, which is the force that drives or ensures the preservation of successful combinations of gene variants. So for example, antibiotic resistance. You have you take your antibiotics, and there might be, just by chance in the population, say a small handful of bacteria in a population of bacteria which have a mutation which confers to them antibiotic resistance. So they happen to be resistant to penicillin or whatever antibiotic it happens to be, and so the penicillin antibiotic then will result in the deaths of all the organisms, the bacteria that are susceptible to the penicillin antibiotic. But then the small handful, the 1 or 2 or 3 or what have you, bacteria which have that particular mutation will not be susceptible to the antibiotic, and so all their neighbors are dying, and now these are the only ones left, and then they become the dominant phenotype, right? So that's how antibiotic resistance occurs, by elimination of those organisms which are no longer able to survive in their environment, and therefore that's what drives this change in alpha-frequencies in the population over time. There's another process which is called genetic drift, which is when you don't have the population sizes that are necessary for natural selection to be effective, and gene variants can become, just by chance, dominant and fixed in the population, just by chance. And so it's a more random process, which can also result in a gene variant becoming dominant in a population, even if it didn't necessarily confer a survival advantage. And then finally, there's what really is thought to drive the variation that natural selection will sift through, which is genetic mutations, which are the copying mistakes in the DNA. And intelligent design really challenges this last point more than the others, which is the sufficiency of random genetic mutations to explain and account for the complexity in design that we see in living systems. So intelligent design, in its purest sense, is perfectly compatible with common ancestry, right? So common ancestry actually wasn't new with Charles Darwin. That goes right back to the ancient Greeks. What was new with Darwin was the idea that natural selection and chance variation could explain the appearance of design without recourse to a designer. But common ancestry is a much more ancient idea. And demonstrating the proposition of hereditary continuity, that all organisms alive today share a common ancestor, even if that's true, it doesn't tell you anything about whether the design that we observe is real design or only apparent design. So that's these issues often get conflated. Now, in terms of detecting design in the world, so I said that intelligent design argues that there were certain features of living systems that are best explained by design. Well, what features of the world would be indicative of design? Well, this is where I would introduce the criteria that were postulated initially by a mathematician and philosopher called William Dembski in his book, The Design Inference, and published by Cambridge University Press. And then there's a sequel to that called No Free Lunch. And basically, specified complexity describes phenomena where two criteria have to be met. The first criterion is that the probability of a phenomenon being produced by chance alone exhausts the available probabilistic resources, right? And I'll explain what I mean by that in a moment. And secondly, the phenomena conforms to some independently specified pattern. So to give an example, imagine you have an ATM machine, right? And we've all used ATM machines. You take your bank card and you push it into the ATM machine and you dial a 4-digit PIN number. And the 4-digit PIN number is specific to your bank card, and presuming that you get it right, the ATM machine will then give you the money that you request to withdraw from your account. Now, there's how many? So if you have a 10-digit PIN pad, so that's the number 0 to 9, and you have a 4-digit PIN number, how many possible PIN numbers are there, right? How many ways are there of dialing a 4-digit number to a 10-digit PIN pad? Well, that would be 10. There's 10 digits, and you would raise that to the 4th power, right? Because you've got 4 different numbers you have to get right. So that's 10 raised to the 4th power, which is 10,000 possible ways of dialing in a 4-digit PIN number. Now, remember that most banks, after you've tried three times and failed, the bank will lock you out of your account, and you'll have to call their hotline to get access to your account again. And so you can think about that in terms of as the probabilistic resources, right? The number of trials, the number of opportunities you have to get it right. So if you only have 3 trials, is it more likely or less likely that by chance alone, if you only have 3 trials, you'll randomly guess the right PIN number for that bank card? Well, obviously it's overwhelmingly more probable that you'll fail and not succeed in guessing the right PIN number. And so the 3 trials correspond to probabilistic resources. And remember that the improbability must always be evaluated with respect to the probabilistic resources, right? So if you have 10,000 trials, then it's much more probable that you'll actually randomly guess the right PIN number. So you always have to bear in mind the number of trials you have to get it right. But supposing that the improbability exhausts the probabilistic resources at one's disposal, then it would satisfy the criteria of complexity or the improbability of the phenomenon. That would be sufficient to qualify as meeting that criterion. Now, when we come to explain or try to account for the specified complexity that we find in the cell, and as you know, the DNA molecule is along the sugar phosphate backbone of the DNA molecule, along the spine of the DNA molecule, are chemical subunits that we represent with the alphabetic characters of A, C, T, and G, right? And the sequential arrangement of those building blocks of these chemical alphabetic characters, if you will, determines in turn the sequential arrangement of the subunits of proteins, which are known as amino acids, right? And there's 20 different types of amino acids, and there's in the DNA there's words which are each of them being 3 letters, which are known as codons. And there's 64 different codons, and each of those codons will specify an amino acid. Sometimes an amino acid might be specified by multiple codons because there's a genetic code redundancy. There's more than 3 times more codons than there are amino acids. So this is so what we see then is this translational system, or this genetic code, this language system. And in every realm of experience, we habitually associate languages and codes and information content with minds, consciousness, and deliberation, right? Intentionality. Now, Stephen Meyer, who is a Cambridge University trained philosopher of science, who now works at the Discovery Institute in Seattle, Washington, he did his PhD thesis trying to develop a method for looking at the evidence of design and actually inferring design as the best explanation for what we see in life. And he drew inspiration from a very famous 19th century geologist who was actually a mentor to Charles Darwin, called Charles Lyell. And he wrote a landmark classic volume in the history of science and his Principles of Geology. And he basically insisted, he put forward this principle of uniformitarianism, which was summarized by the diction that the present is the key to the past. And he basically argued, if you want to explain changes in the Earth's surface in the deep past, we should let our present experience of the cause and effect structure of the world guide that search for the best explanation. And so this is now known as the abductive method, or the inference to the best explanation for multiple competing hypotheses. And so Stephen Meyer basically took the idea of Charles Lyell and applied it to, and by the way, that idea was instrumental in the way that Charles Darwin formulated his theory of evolution by natural selection. It's using the same sort of methodology of investigating the past. So Meyer took that basic model, that basic historical method, the abductive method, and applied it to our question here. When we're looking at biological information content, we know of only one type of cause, one category of explanation, which is known in every realm of experience to produce this type of effect, and that is intelligence. Intelligence alone is habitually associated with information, especially in a digital form. And so when we see information present in the DNA molecule, encoded in digital form, then the best candidate explanation for that is that it too arose by virtue of an intelligent cause. So that's basically the design argument in a nutshell.
Yeah, that even though I am a layman when it comes to things such as science, that was fairly easy to understand. And this is actually, you brought up Stephen Meyer, and for those who are not aware, yes, he is a philosopher of science, but that's the same argument he brings up in his book Darwin's Doubt, which has been a New York Times #1 bestseller for quite a while now, and in which he equates the DNA code, or the genetic sequence, to a computer code. And this is also what the computer scientist David Galertner at MIT, I believe, or Yale, he also makes the same equivalency between computer code and DNA code. And you touched on Darwin as you were talking about intelligent design. And today, this is David Berlinski's criticism of Darwinism today, otherwise known as neo-Darwinism, is that neo-Darwinism has now become a dogma in science, in essence, that people take Darwin's theory to be the be-all, end-all, that there is no competing theories when it comes to evolution and how species have evolved over time. What would you say would be the problems associated with neo-Darwinism today? I know you brought up you alluded to the inefficiencies of mutations in what you were just saying, but what are some of the big problems associated with neo-Darwinism today?
Absolutely. So there's many problems associated with the neo-Darwinian evolutionary theory. I'll just give a few. So as I mentioned, the subunits of proteins are amino acids, and the sequential arrangement of those amino acids is determined by the sequential arrangement of the coding parts of DNA, which code for those proteins, code for the amino acids that form the subunits of proteins. And the sequential arrangement of the amino acids that make up the proteins, by virtue of the properties of the side chains of the respective amino acids, will determine how a protein will collapse into a three-dimensional structure. And the way that a protein folds into a three-dimensional structure, in turn, is responsible for how that protein will behave in the cell. Either it might be involved in it might be an enzyme that forms a part of some biochemical pathway, or it might be a component of a molecular machine in the cell. And it turns out that creating new proteins by chance and necessity is highly problematic. So, for example, I mentioned earlier that there were 20 different amino acids that are used in cellular proteins, and that means that for a monocular length protein of, say, 150 amino acids, there are how many possible ways of arranging those amino acids? Well, that would be 20 raised to the power of 150. So that's 20 times 20 times 20 times 20, all the way up to 150, right? So that's a huge combinatorial space. Just to put that in context, there's about 10 to the power of 65 atoms in the galaxy, right? There's about 10 to the power of 80 atoms in the known universe. So, there's more ways of arranging a molecular-length protein of 150 amino acids, way more ways than there are subatomic particles in the known universe. So it's just huge combinatorial space, huge number of ways of arranging those amino acids. Now we know that many of those are functional, and many of those are gibberish. Just as in the English language, there's far more ways of writing rubbish than there are ways of writing meaningful sentences, right?
Right.
There's far more numerous ways of just writing things that don't make any sense than there are ways of putting together structured sentences and paragraphs and so forth. And the same thing turns out to be true in life. So the question that we can ask is, how prevalent are functional protein folds in combinatorial space? And here I think that Darwinism and design make competing predictions. So Darwinism would predict that you would find a relative abundance of protein folds within that vast array of combinatorial space. That is, amino acid sequences that form stable and functional folds. Because then it would seem quite reasonable and plausible that by a blind, unguided, chance, random process, that you would be able to stumble upon the functional folds by just a blind search through that sequence space. However, if it turned out that the preponderance of functional and stable protein structures within that vast array of combinatorial possibilities was astronomically small, then how would you by chance alone, because bear in mind natural selection can only preserve function once it's been found by chance, right? How would you find by chance alone the functional sequences within the scope of the probable state resources that are afforded by the universe? So this question has been taken up and there's actually a number of papers that address this question. The most well-known, or the most famous of which, is Dr. Doug Axe in his book, in his journal paper published in the Journal of Molecular Biology, called "Estimating the Prevalence of Protein Sequences Adopting Functional Enzyme Folds." And he basically examined a domain of a protein from an enzyme which confers antibiotic resistance against penicillin, called beta-lactamase. And it's about 150 amino acids in length. And I won't get into the technical details, but basically he estimated that the ratio of stable and functional protein structures within combinatorial space is about 1 in 10 raised to the 74th power, right? Now, as I said earlier, to put that in context, there's only 10 to 65 atoms in the galaxy, about 10 to 80 atoms in the known universe. So that's a huge, that's a very astronomically small rarity. And so you're talking about searching for a tiny, tiny needle in an enormous haystack and having a very limited time to do so. Bear in mind that the number of seconds that have elapsed since the Big Bang is only 10 to the 16 seconds. Right. So and Doug Axe, I mean, he's the most well-known publisher on this topic, but and he's published other papers making a similar argument as well. So this isn't the only research that he's published on this, but there's other papers that actually come to similar conclusions. You can find other papers that make estimates within 10 to 60 to 10 to the 80 range. So it seems to be the case that the prevalence of functional protein folds within combinatorial space is astronomically small. And so how by natural selection would you be able to find the protein structures just by chance? And so it presents this probable state resource problem. So there's approximately 10 to the 80 elementary particles in the observable universe, and as I said, since the Big Bang, roughly 10 to 16 seconds have elapsed. And the time it takes light to traverse the smallest distance is 10 to the minus 43 seconds. And so by multiplying together these three relevant factors, we can determine that the maximum number of events that could have taken place in the observable universe since its origin is 10 raised to the 139th power. And so when so locating the functional sequences within the vastity of combinatorial possibilities by a random search is like finding a tiny needle in this enormous haystack.
Right. Yeah.
And it's if you and when you consider the fact that to get the origins of life, you don't just need one protein or two proteins or 10 proteins, but you need two or 300 proteins at a bare minimum to even get life off the ground. It's just way off the page. And furthermore, when molecular machines require multiple cooperating proteins in order to perform their function, the probabilities multiply exponentially, right? So, for example, take the DNA replisome, the replication machinery that replicates DNA, and there's many proteins that are involved there in carrying out the replication process, and each of these proteins has to be specifically crafted in order to cooperate together in order to perform an overall higher level objective. And so the probabilities just multiply exponentially as you need these proteins that perform mutually cooperative roles. And in fact, Charles Darwin, in his The Origin of Species, published in 1859, put forward a test that could potentially falsify his theory of evolution by natural selection. He wrote that if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. And this challenge was taken up by a biochemist at Lehigh University called Michael Behe in his book Darwin's Black Box, published in 1996. And he coined this term irreducible complexity. And he said, by irreducible complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. So he was basically talking about systems where if you have this higher level objective and you have multiple subfunctions that are contributing to that higher level objective, if you remove any one of those components that are performing those subfunctions, then you'll lose that higher level objective. And so how would you, by a process of natural selection and chance variation or slight successive modification, build up to a higher level objective without having the functionality all along the way? Because if you remove one of the subfunctions that's contributing to that higher level objective, then you don't have a system that works half as well as it used to, or a third as well as it used to, but it's broken. So he gave the example of a mousetrap, which is a well-known device from our everyday experience, which is comprised of a number of components. It's got a platform, it's got a spring, it's got a hammer and a hold-down bar, a catch and stables, each of which is contributing to this higher level objective of catching mice. And in fact, even with those components, the trap might still not work if the holding bar was too short or the spring too lightweight. So each of the components has to be matched one for the other. And if you remove the spring, for example, you remove the hold-down bar, then you don't have a trap that works half as well as it used to, or a quarter as well as it used to, but it's just broken. And when we look at biological systems, we see that many such systems that bear this property of irreducible complexity are found there as well. So I mentioned earlier the example of DNA replication machinery. And if we look at just some of the components that are necessary, so you need the DNA polymerase, for example, which is the enzyme which replicates each of the two strands of the DNA double helix. And without that protein, no replication can take place at all. The RNA primers would be laid down at the organ of replication. These are required to kickstart the process of DNA replication because the polymerase isn't able to start on its own, but then there would be no subsequent replication. If we didn't have the DNA ligase, which linked the DNA fragments that are produced on the lagging strand, because the lagging strand of the DNA, there's two strands of DNA, it's a double helix, right, and they're bi-oriented. So one of the strands actually has to be copied backwards, which is done by synthesizing what's called RNA primers, and then replicating from those, creating what are called Okazaki fragments. And these have to be linked with what's called a DNA ligase. So without that, then the newly replicated DNA strands would just remain as fragments, You also need a sliding clamp to clamp the DNA polymerase onto the DNA, and without that, then the DNA polymerase would just frequently fall off the DNA template, and it wouldn't be possible to continue replicating the DNA. It would just be too slow. If you didn't have and you also need a clamp loader as well, in order for that to work. If you didn't have the RNA excision enzymes, and the RNA fragments would remain attached to the newly replicated DNA fragments, because the RNA fragments, the RNA primers have to be removed. So you know, that wouldn't take place. You need the DNA helicase, which is the enzyme responsible for unwinding the DNA. Double helix to prepare for copying, and of course, you also have single-stranded binding proteins, which bind to the two strands of the DNA to prevent them from re-annealing during copying. And you also need the primase, which without that, then you wouldn't be able to have the RNA primers to begin. You would be able to lay down the RNA primers on either the leading or the lagging strand, because the RNA primers have to be laid down before the polymerase can replicate the DNA. And so DNA replication in that situation wouldn't be able to begin at all. And so you just look at all these components, all these protein components, which need to be present in order for evolution, in order for DNA replication to work. And if any of those pieces was missing, and that's just very simplified summary, there's many other components that are necessary as well, then you wouldn't have a DNA replication system that works half as well as it used to, but would be broken. And so that's I think a significant problem. And this is what I call actually an example of irreducible complexity on steroids, because not only is it irreducible complex, but for natural selection to even work, you need a self-replicating system, right? You need replication, because how can natural selection select for the fittest organisms if they're not replicated, right? So you don't have new generations of organisms that are able, and so DNA replication is absolutely fundamental for that process. And so you cannot even invoke natural selection to explain something like DNA replication without assuming the existence of the very thing you're trying to explain. So there's many other problems we can talk about in regards to challenges to evolution, but there's just a couple.
Definitely. And basically what I'm getting from that, and correct me if I'm wrong, the argument or the problem with neo-Darwinism today is just that you just need so many things to come into place at the right time, at the right place, all together. And all those things and the chances of those things coming together is almost infinitesimally improbable.
Exactly.
Right. And I think when it comes to neo-Darwinism, what do you think the problem is, especially with other evolutionary biologists, your colleagues in the field? What do you think is the problem with them having trouble to accept this theory of intelligent design? Would you say that it's more of a problem of simply accepting that there has to be some designer, or is it just pure scientific dogmatism?
Yeah.
And once again, this is Berlinski's critique of the field of evolutionary biology today, in the sense that nobody's able to accept that there are flaws in Darwin's theory.
Yeah, that's a great question, and I think for many scientists that I've talked with, they are not even familiar with the basic points. They're not even familiar with the basic issues that are live in the debate, and so they're ignorant, and that's certainly true of most scientists I've interacted with, because most of them aren't evolutionary biologists. It's actually their fields of specialization. Because in science, you're very focused on your field, and you don't tend to, at least for the most part, read much outside of that. And so most biologists kind of just take it on authority that evolution is well established. And even among people who are evolutionary biologists, I've talked to people who are well established evolutionary biologists who aren't even familiar with the criticisms raised by proponents of intelligent design. And it's quite sad and often they have misconceptions about what we're arguing because they've only read about it from our critics, right? And in some other cases, in some cases, I think that there's metaphysical commitments that are also live in this discussion as well. So, for example, Richard Lewontin, who's an evolutionary biologist, he writes, and I'm quoting, he says, "Our willingness to accept scientific claims that are against common sense is the key to an understanding of the real struggle between science and the supernatural. We take the side of science in spite of the patent absurdity of some of its constructs, in spite of its failure to fulfill many of its extravagant promises of health and life, in spite of the tolerance of the scientific community for unsubstantiated just so stories, because we have a prior commitment to materialism. It's not that the methods and institutions of science can somehow compel us to accept a material explanation of the phenomenal world, but on the contrary, that we are forced by our a priori adherence to material causes to create an apparatus of investigation and a set of concepts that produce material explanations, no matter how counterintuitive, no matter how mystifying to the uninitiated. Moreover, that materialism is absolute, for we cannot allow a divine foot in the door. The eminent Kant scholar Louis Beck used to say that anyone who could believe in God could believe in anything. To appeal to an omnipotent deity is to allow that at any moment the regularities of nature may be ruptured, that miracles may happen." So there you see Richard Lewontin, a very prolific, prestigious evolutionary biologist, admitting that really the reason that he rejects design explanations is not because of somehow being compelled by the scientific evidence, but because of a methodological commitment, a commitment to material causality,
Right.
a commitment to materialism. And therefore, it's a methodological bias, it's an anti-supernatural bias of methodological naturalism, which I actually think is irrational and anti-intellectual.
Right. Now, I wanted to try and poke a few holes in the argument for intelligent design, but before I do so, you brought up the complexity of amino acids and the cell. Are there any other aspects that you could point to that would be evident of intelligent design that are seemingly so complex that it's very improbable that these things would have developed over time?
You mean in terms of other irreducible complex systems?
Right.
Yeah, so just bear in mind, it's not so much the quantity of the complexity that's the issue, but the quality of the complexity, right? So there's a lot of complex things that can be produced by physical unguided processes, right? But it's the quality of the complexity, and the reason, by the way, that irreducible complexity is evidence, I would argue, of design, is that explaining the origins of irreducible complex features, that is where you have a high level objective that's contributed by numerous subfunctions. That sort of phenomena seems to require a cause that possesses foresight, and indeed only intelligence can visualize complexity and bring together everything that's necessary to actualize a complex endpoint. So I'll give another example of a system. So take, for instance, bacterial cell division. And bacterial cells divide by a process known as binary fission. If you have a rod-shaped bacterial cell, it will elongate to about double its original length, and then you'll have a polymerization around the division septum of a protein called FitZ, which forms what's called the FitZ ring. And that will constrict and basically cause the cell to bifurcate in the middle and differentiate into two daughter cells. Now, what's interesting, there's a lot of molecular complexity we could discuss there, and actually there's, I think, multiple examples of irreducible complexity just associated with the system. But let me just focus let me just focus here on just one of those. That is that when the bacterial cell is elongating to prepare for division, you have to literally sever the peptidoglycan cell wall along the bacterial cell. And this is done by proteins known as autolysins, which have to be very carefully controlled and regulated, because if they get out of control, then it can cause programmed cell death, which results in the cell committing suicide, essentially, or killing itself, which is not what you want. So you have so bacterial cell division then requires this growth of the peptidoglycan cell wall as the bacterial cell elongates, and that process occurs by the severing of the peptidoglycan backbone or the cell wall, and the synthesis of new cell wall material. And so you've got these autolysins, which are severing the cell wall, and you have to coordinate that with the insertion of new cell wall material. And there's a process known, there's a protein called bactoprenol, which shuttles the peptidoglycan precursors across the cell membrane to the cell wall. And there's a process known as transpeptidation or peptidoglycan cross-linking, which is accomplished by proteins known as penicillin binding proteins. And these penicillin binding proteins, as their name implies, is the target for penicillin antibiotic, because penicillin antibiotic disrupts this transpeptidation process, which really shows you the importance and crucial nature of this process. Because without it, if you don't if you don't carry out the peptidoglycan cross-linking, then because of you've broken the cell wall because of the osmotic pressure, basically the cell will burst, right, and will die. And so you have this process for ensuring the reconstruction of the cell wall to prevent that from happening, which has to be done in a coordinated way with the breaking of the cell wall. Now consider the following two observations. So number one, critical to the elongation process is the severing of the peptidoglycan cell wall by the autolysins I talked about. And secondly, critical to the cell viability is the resynthesis of the peptidoglycan cell wall. So breaking the cell wall can serve no adaptive utility until a mechanism has arisen for the simultaneous integration of peptidoglycan precursors. And without the latter mechanism, the cell is rendered non-viable. And so that's an irreducible complex system, because if you have the mechanism for breaking the cell wall but not the mechanism for rebuilding the cell wall, then your cell is going to die. You need both to work together in a coordinated way in order for cell division to take place in bacterial cells. So that's another example, I think, of an irreducible complex system.
Awesome. Once again, it's clear neo-Darwinism or neo-Darwinian evolution has its problems. So let's try and look at some of the quote-unquote problems with the idea of intelligent design, and let's see how it holds up to these objections. First objection would be an objection that I could imagine coming from Richard Dawkins, and that objection being that this argument of intelligent design seems to be another classic example of the God of the gaps argument, in the sense that we see something, and that something just seems so impossibly outrageous to us that it must be God. Why can't we just say intelligent design is just a product of randomness? Why do we have to assert some level of transcendental intelligence to the origins of species?
Right. So the way that intelligent design is typically argued, as I said, is as an inference to the best explanation. So we know that intelligence is the one known type of cause or category of explanation that's known in every realm of experience to produce information content and these sorts of higher level objectives I talked about, and systems that require a foresight to bring about, such as developmental trajectory, for example, where you have to visualize a complex to bring about a process like that, where you have this end goal in mind. You have to be able to visualize a complex end goal and bring things together to actualize that end goal. So it's an inference to the best explanation. It's arguing that in every realm of experience, these sorts of phenomena are habitually associated with design. Another way you could parse the argument out is in terms of a Bayesian approach, which is to say that the phenomena that we observe in biology is more probable on the hypothesis of design than it would be on its falsehood. And we know that designing agents often produce information content and high-level objectives and so forth, or functional coherence, if you will. And so it's not at all improbable that if life is the product of design, then it will contain these sorts of features. But it's wildly improbable on the hypothesis that no design is involved, that those features would exist. So there's another way of parsing out the argument.
Right.
And so whatever else might be wrong with the design argument is not wrong because it's a God of the gaps argument. So I think that objection is wrong-headed.
Right. And we're just hitting our time here. I'm going to give you one more objection, and this is also another objection that is pretty common throughout. I wouldn't say necessarily academia, but I would say maybe internet atheists, being that, you know, intelligent design sort of implies to people that everything has a purpose in the body. So how does intelligent design explain things such as vestigial structures, such as the idea of wisdom teeth and the jaw having too much teeth, or the idea of the birth canal of the female being too small to allow for the head of a baby, for example? Like there are many things in which, you know, we see that don't necessarily have any use, or there are things that are sort of imperfect that we would, you know, consider to be as such.
Yeah, that's a great question, and one that often comes up. I tend to, my personal preferred way of parsing out the argument for design is along the lines of the Bayesian approach that I described, where we would argue that the features that we observe in life is more probable on the hypothesis of design than on its falsehood. Now, when we're evaluating things in terms of a Bayesian approach, and let me just summarize what a Bayesian approach is. So for a Bayesian, a particular data point or a fact is evidence for your hypothesis if and only if the probability of that evidence given that hypothesis is greater than the probability of that same evidence existing given the falsity of that hypothesis. So we're measuring evidence in terms of this likelihood ratio, the probability of the evidence existing given your hypothesis is true versus the probability of that same evidence existing given the hypothesis is false. Now, an important thing to bear in mind is that the probability of the evidence on the hypothesis, on the assumption of the hypothesis, doesn't need to be high in order for you to have evidence for your hypothesis. It only needs to be higher than the probability of that evidence only needs to be higher given the hypothesis and given its falsity. So even if it's improbable given the hypothesis, as long as it's more probable on the hypothesis than given the falsity of the hypothesis, it still contributes evidence to your case. So even if it is the case that the jaw has too much teeth, or the birth canal of the female is too small and the designer wouldn't have done it that way. Let's just say, save and sake argument that it's unlikely, it's improbable that a designer would have designed those things the way he did. It's still more probable that a designer was responsible than the falsity of the hypothesis that no designer was responsible, because then it's wildly more improbable. So that would be one of the responses I'd make to that. And also, there could be explanations. There could be purposes that we just don't know about yet. So it's kind of a converse God of the gaps fallacy or Darwin of the gaps fallacy. Just because we don't know why the designer did it that way, therefore we'll just plug in Darwin to explain that of which we are ignorant, use it as a placeholder for that which we don't know. And furthermore, there are examples of features that were once thought to be poorly designed, which turned out upon further inspection to be well designed. And actually we discovered reasons. And so actually intelligent design at that point has a heuristic value of actually driving further scientific discovery, because it encourages us to look for design reasons why structures are designed the way they are, when actually Darwinism would encourage us to give up and suggest that actually it's just there is no design reason. And furthermore, there might be developmental constraints. There might be even if you are some ID proponents like Michael Behe or Michael Denton and others even accept common ancestry and would argue that there's design irrespective of the issue of just being descended from a universal common ancestor. And you could have like the case of the recurrent laryngeal nerve, for example. Perhaps that's just a byproduct of its evolutionary heritage, even though it's a directed evolutionary heritage. So there are explanations of that which are perfectly consistent with design anyway. So I actually don't find that a very convincing objection to design.
Yeah, me as well. Only you also brought up the idea of, you know, flipping the God of the gaps argument on its head. This can also be applied to the philosophical problem of evil, or more so the probabilistic problem of evil, in the sense that, even if you can point to evil in the world, right, the burden of proof on the naysayer is so high that they have to prove a logical reason as to why God or a greater being has no reason to permit such evil. And it seems to be that the same argument can also be made here, in the sense that the burden of proof is so high for the atheists in the case of evolution, that they have to prove that an intelligent designer has no good reason to have things such as vestigial structures or parts to the human body that seemingly have no reason to exist now, but in the future, right, perhaps we may find that explanation. And for those, he was talking, Dr. McLatchie was talking about Bayesian probability. For those of you who do not know what it is, it's actually a pretty popular form of propositional logic, or it's used in propositional logic a lot, especially in apologetics. And if you guys really want to learn more about Bayesian probability, there's a good article in it in the Plato Encyclopedia of Philosophy by Stanford, I believe. So we just surpassed a little over the hour mark, but I'm sure many people will have questions about intelligent design, Dr. McLatchie. So what are some resources you can point people towards if they want to learn more? Or perhaps if people have been feeling convinced, what are some good sources to expand their knowledge or to strengthen their knowledge when it comes to apologetics and evolutionary biology and things of the sort?
Sure. So the website I'd recommend most is evolutionnews.org, which is the blog of Discovery Institute. I write there quite a bit, and there's other writers there as well, making a scientific case for design and raising critiques of the standard neo-Darwinian evolutionary scenario. In terms of books I would recommend, I recommend Signature of the Cell by Stephen Meyer, which was published, I think, in 2009. And also the subsequent book called Darwin's Doubt, which is on the Cambrian explosion of life, and makes a case that there are certain features that basically invent the history of life, where many of the animal body plans or the phyla appear very abruptly within a very narrow window of geological time. There's a Cambrian explosion, and he argues that there are certain features of that explosion which point to design. And he's coming out with a new book, actually, called The Return of the God Hypothesis, which is probably coming out next year. And Michael Behe has 3 books as well. He's a biochemist at Lehigh University. He pioneered the argument from irreducible complexity. His first book, published in 1996, was called Darwin's Black Box. Then I believe in 2006 or 2007, he published a book called The Edge of Evolution. And then in 2019, he published his 3rd book, which was called Darwin Devolves. So I highly recommend those. Also, one other book I'll recommend is Dr. Douglas Axe's book, Undeniable. He's a molecular biologist who now works at Biola University in Los Angeles. And he's the guy that I mentioned being responsible for the research on the rarity of protein folds within that combinatorial space, his research to beta-lactamase. He wrote a book which is aimed at a general audience called Undeniable, which basically articulates that argument in easy to understand language. So these are a few resources I'd recommend.
And Dr. McLatchie, why don't you also tell the listeners where they can learn more about you, or where they can find you more on social media, or maybe on the internet for perhaps more lectures on intelligent design if they also want to learn more?
Sure. So my website is jonathanmcclatchie.com. You can find I've got a YouTube channel called Dr. Jonathan McLatchie, and I also have another YouTube channel called Apologetics Academy. And that should be my website is a good launching pad for all my other stuff that I'm doing. So that's probably the best place to find my resources.
Awesome. And jonathanmcclatchie.com, right?
Yeah, that's correct. Mm-hmm.
Right. All right. And okay, guys, you heard it, jonathanmcclatchie.com, if you guys want to learn more about Dr. McLatchie and what he's doing and his future projects. If you guys have any questions, or if you guys want to get in contact with Dr. McLatchie, or if you guys want to learn more, I'm sure there are sources on his website for you guys to do such. And also, Dr. McLatchie recommended a bunch of good books. I personally own Darwin's Doubt by Stephen Meyer. I haven't finished the whole book yet, but I intend to do so after this enlightening discussion with Dr. McLatchie. And that will do it for today's episode. I want to thank you guys so much for listening in to The Prodigal Believer. This is PB, and I'm here with my special guest, Dr. Jonathan McLatchie. Thank you guys so much for listening in, and we'll see you next episode.